Good morning, everybody, and thank you for joining. My name is Faith Dukes. I'm Director of STEM Education and Workforce Programs here at Lawrence Berkeley National Laboratory. Today's career talk and research talks today will be focused on the biotech tools transforming research. We have four wonderful speakers that will introduce themselves in the first half of our session, talk a little bit about their background, and then share a little bit about the work that they do and the tools they use in their research. And then we will transition to a panel discussion. So get ready to put your questions in the chat for this discussion. We look forward to hearing your questions and hearing from our speakers. Today, the first speaker we'll start off with is Shreya Garg from Stanford University, who will tell us a little bit about her background as well as her work at Stanford University. So Shreya, feel free. I'm going to stop sharing and you can share your slides.
Yeah, thank you so much for the introduction, Faith. All right. So, hi everyone. My name is Shreya. I'm a first-year PhD student in the bioengineering department at Stanford University, and I'm really excited to talk to you all about some of the biotechnology tools that I've used in my work today. A bit of background, I originally grew up in the Bay Area in California, did my undergraduate work in bioengineering and science communication. And throughout college, I hopped around from field to field until I settled into where I am now, tried out everything from pharmaceutical design, diagnostics, medical devices, 3D printing, all of which really cultivated an interest in material properties and product design. And so that led me to where I am now for my PhD in Dr. Vayu Hilmaini's lab at Stanford, where we study genetic engineering in fungi. And my ultimate goal down the line will be to study how we can manipulate fungal textures and fibers to create materials, textiles, and more. And so here are just a couple of pictures of folks in the lab. We operate anywhere from in the lab to the kitchen with some partners in NASA. So it's a really cool group of people, and I'm really excited to be working with them. So I'll start with a brief overview of Vayu's lab before I dive into my own work. The lab focuses on the genetic engineering of fungi, exploring many dimensions such as plant-fungi interactions, fungi-based textiles, new fungally derived foods, how fungi can convert food waste into useful products, and a lot more. And you can get a sense of just how versatile the fungal world is just by looking at them. The pictures here are just some of the fungi found in the Bay Area in California. And already you can see how diverse they are in color, texture, shape, aesthetics, and more. But less visibly noticeable, but also super crucial, are how they vary in the proteins or the metabolites that they secrete, such as antivirals or antibiotics. If we can identify the genetic sequences and pathways responsible for producing these unique traits, we can then use them in our engineering applications. For example, synthetic dyes in the textile and fashion industry contribute to around 20% of industrial water pollution. And just by looking at the colors in these images, we can see that the array of pigments produced by fungi. So what if we could isolate these pigment genes, amplify them, and then use fungi to produce our dyes more sustainably? That's just like one of the questions that we're hoping to answer. Lab members are already exploring this question, as well as how we can use fungi to keep soil ecosystems healthy, how we can use traditional fungal fermentation pathways to ferment food waste back into food, and a lot more. So now let me get into just what I have personally worked on in the lab. And to preface, I just joined the lab a few weeks ago. So I'll give you more of a general overview of the project as it is still very much ongoing. So currently, if you want to engineer a strain of fungi, you need to individually create a mutant strain, grow it up in an isolated environment, such as a petri dish, and then analyze whether it does what you hoped it would do. And this process can take weeks to months and use many lab resources, all to test a single mutant, let alone the fact that fungi are not that well understood in the lab right now. So there's many unanswered questions for how even to genetically engineer them and those types of species that we are able to work with. And there are thousands of variants that we would ultimately want to test. So this bottleneck in the current process makes bulk testing very infeasible. So what if we could instead batch test hundreds or maybe even thousands of mutants at a time? that's where the biotechnology tools of synthetic biology and microfluidics come in. Our goal with this project that I'm going to describe to you now was to develop a system that was high throughput, or in other words, could test a high quantity of variants all in one go. So the first step was to develop our high throughput platform. For this, we use something called microfluidics, a tool that is exactly what it sounds like, where we use devices that manipulate small volumes of fluids. So here we specifically use a tool called droplet microfluidics, which I'm going to explain using this diagram here. So we can insert a liquid solution of a bunch of fungal spores, run them through this device, this microfluidics device, where they will then be encapsulated in droplets coated by a hard shell that traps them in their droplets. So we start out with a very dilute solution of spores, run them through this device, and because of their very small concentration, it's unlikely that we have more than one spore in each droplet that comes out of the device. All right. And so if you were to put a bunch of different spores on a petri dish, they would, as they're growing up, definitely contaminate each other. And once they grew up, you wouldn't really know which one was which. But because of this microfluidics platform, we can still grow a bunch of spores together in a single, maybe like liquid flask, because they're all isolated in those hard shell coated droplets. And so we no longer have to use several petri dishes, flasks, nutrient solutions, and more for each individual spore. And then, of course, we'd have to do that for multiple replicates. Creating these droplets just takes a few hours, and growing up fungi in small droplets takes just a few days, ultimately saving us months of time to test our variants. Okay, so now we have our platform. Next comes the actual engineering of the spores using the next tool, synthetic biology, which is just the general field of engineering living organisms. Our group wanted to create variants that express the green fluorescent protein, or GFP. And this would basically just make the spores glow green under a fluorescent microscope. And we wanted specifically to vary the spores by having them vary in how strongly that GFP was expressed. And so we can do this by controlling the promoter sequence that controls the GFP expression. And so here's like the symbol that we'd use for a promoter. A strong promoter would be able to induce a lot of GFP, making our fungi very fluorescent, whereas some weak promoter would basically just produce a very tiny bit of GFP, leading to a much fainter glow. And we can engineer several strains. So we can engineer several strains of our fungi with different promoters and then the same GFP gene, capture all of those individually modified spores into droplets, grow them up in these droplets, and then see how fluorescent each droplet is. A very fluorescent droplet, again, would have something like a strong promoter. And then after we've grown them up, we can get the genetic sequence of the fungi growing inside and then determine what promoters led to what level of fluorescence. And testing a bunch of promoters might not seem very useful in the context of, making fungi glow at various intensities, but you can imagine other a lot more useful applications that are a lot more important. For example, what if we wanted to use fungi to mass produce pharmaceuticals? We might want a strong promoter to get a very high yield. Or if we wanted to create a new fungally derived food with a very subtle flavoring protein, we might want to use a weak promoter for that protein. And so just to provide some visuals of what this looks like, here are some droplets with spores. And then after a few hours or maybe about a day, they grow up to about this size. And let me see if I can get this video of the droplet generation process going. Yeah, so here is what the microfluidics process looks like. This is under a microscope, and this is what we're seeing through that microscope. You have the fungal spores basically flowing through this device, and the hard shell coating that forms around those droplets. And you can see that what comes out of the device are these droplets, and all of them will have either 1 or 0 spores inside of them. So super cool. And then over here, we have some images of fungi in those droplets under a regular light, and then also when they're fluorescing under a fluorescent microscope, which ultimately shows us that our synthetic biology steps were also a success. And so currently we're still in the process of optimizing our droplet generation and creating the variants that we want to test. As I mentioned, we're just getting this project going. The lab is very new, but we're also very excited to see what comes of this. So yeah, I hope you guys enjoyed hearing about how genetic engineering can be applied to fungi. As I mentioned before, I've worked with a lot of other tools like 3D printing. So if you have questions, feel free to ask. And I definitely don't have the same depth in research and experience as some of the folks that will be talking today, but it wasn't too long ago that I was confronted with some of the same questions that you might be facing, such as figuring out what path to pick or narrowing down research interests. So very happy to answer any of those questions too. But yeah, thank you so much for listening.
Thank you, Shreya. Also in the chat, I'm going to put the link to your group's lab so they can learn more about the research that you all do. So again, as Shreya said, you all have an opportunity to ask her questions later on. I'm going to ask the next speaker to come up and share their screen. So Maureen Berg is a research scientist here at Lawrence Berkeley National Laboratory in our Joint Genome Institute. And we look forward to hearing more about, again, her trajectory as well as her work here at the lab.
Okay, sorry, let me unmute. Hi, everyone. So funny enough, I will also be talking a little bit about microfluidics, but in a different capacity. But before I get into that, I'm Maureen Berg. I'm a staff scientist at the Joint Genome Institute, which is here at Berkeley Lab. And so a little bit of background, because I've had a slightly variable research experience. So initially, and I was born and raised in Southwest Ohio. And I also went to high school there. You can tell this is from the 2000s because of the low quality digital camera. And I did my undergraduate degree and research at the University of Dayton in Ohio. And this was an ecology and entomology lab. So we studied insects and we also did some microbiology, but this was primarily a fieldwork lab. So I did a lot of field work. And I learned that I don't like field work, but it was still a good experience. We worked on some freshwater ecology, streams, rivers, lakes. And the project I worked the most on was decomposition ecology. So I essentially was out in the woods watching pigs rot and collecting insects and microbes associated with that process. So yeah, this was a great experience because I got a lot of field work and some ecology experience, but for my graduate work, I wanted to not do field work and to get a little bit more genetic and molecular biology experience. And so I did my PhD at Berkeley, and the main kind of themes of the work I did was kind of host microbe interactions. So specifically, I don't know if you can see my cursor, but I worked with a model organism nematode called C. elegans. And the benefit of working with model organisms like this worm is that they are, you can manipulate them easily, like they're like genetics. You can make mutants of them. This worm is very clear, so you can make them fluoresce green or red when they have different processes going on. They're very easy to manipulate and engineer, essentially. And so I was looking at the bacteria that live in the gut of them and how those bacteria interact with their worm host. And so this was a good experience because, like I said, I got out of the field and doing more kind of molecular and genetic work. I also did kind of a side project on plant microbes interactions, asking the same questions, but just in a different system, which is beneficial because, you kind of get a little bit more experience working with different systems. And so for my postdoc, I was a postdoc at the Joint Genome Institute. And I guess if you don't know what a postdoc is, it's, what you do after you get your PhD and you just do research, but you're not paid very much. But they're usually kind of good experiences to, if you want to kind of go into a slightly different field or maybe learn a different skill set. They're kind of good ways to train for that. So I worked in, I'm still in the same group, but I was working in a virus group. And so I was looking at bacteria virus interactions in freshwater lakes. And so I did a lot of lab work and some computational work. And one of the reasons why I really liked this position for after my PhD is because I was doing more computational work as well. So I wanted to kind of expand my skill set even more outside of just lab work. And so right now, I'm a staff scientist still in the same group. And I still work on bacteria virus interactions in different systems, freshwater lakes, soils, everything. But I also now have a new project where we've been working on more kind of technology and tool development, which is what I'll talk about more today. Um, and so that's kind of the really high level overview. But the kind of the theme is just I wanted to, expand my skill set and have kind of a well-rounded, research experience. So yeah, so like I said, I am talking a little bit about microfluidics, but in a different capacity. Um, the kind of new project, well, I guess it's a year old, but somewhat new technology that the group I've been working with have been working on is this semi-permeable capsule technology. So it's basically just a way that we can better manipulate individual cells. So before kind of getting into the details of it, like this is what it looks like. The picture on the left is the setup of all the syringe pumps to put all the fluids. And then the picture on the right is the actual microfluidic chip showing all the tubing going into it. And so hopefully it's not too overwhelming, but basically how they're made is we take, you can use any cell, but I work with bacteria, so I'm talking about bacteria. I take bacterial cells, mix them with these polymers, and put them in these little droplets. And when they're in the droplets, they're kind of like this liquidy mixture. And then you shine a special light on them, and the mixture turns into like a hard shell. So basically, it's like a hard shell that traps a bacterial cell in it. But this hard shell is semi-permeable. So you can get different enzymes and buffers and things to go in it, but everything with the bacteria is trapped in there. So it's a way to kind of manipulate the cell without, doing everything in like a single well or something. So you kind of do it all in a bulk situation. So this is like a low quality video of what it actually looks like running. So you have the different channels coming in. As Freya showed you before, a lot of microfluidic chips kind of look somewhat similar, but that's what it would look like. So in terms of applications, there are a lot of different applications we're working on right now, but I just picked one just to kind of briefly go through as a kind of conceptual thing since I was talking about viruses, and that's a lot of the work been working on. So as a super, super fast background. In my postdoc, I was looking at viruses in freshwater lakes. So it's how do we look at that? So but you can basically how we were doing it is basically you can take some water and you can just sequence all of the DNA in there and hope you'll get a bunch of viral genomes and you can say, oh, these are all the viral genomes I found. Cool, this is what's in here. However, viruses, these viruses infect bacteria. That's kind of their whole deal. That's their, role in the ecosystem. And that information does not come from this standard sequencing of just getting all the genomes. And so that's kind of a key piece that we really want to be able to develop a good method to kind of link a bacterial cell and the virus that infects it to understand the ecology side better. And so going back to these SPCs, so as Shreya said, well, she had spores, but I have cells, but you have at most one cell in a little capsule. And so the idea is you put all your cells in your capsules, you make them, they're in these little cages, you're all good to go. And then using different molecular techniques, we can barcode the genes of the bacteria and any virus that is infecting that bacteria at the same time. And so these barcodes are SPC specific. So I'll show a diagram in the next slide in a minute, but basically whatever's in a single little capsule cage gets the same barcode and it's special to that. So you can kind of link these back and later on. So I have a little diagram to explain that. So essentially on the top, you have your bacterial genomes with their special little barcodes that we engineered. And then that are SPC capsule specific. And then the bottom, the blue, you have your viral genomes that also have the barcode. And so you sequence everything, everything's barcoded. And when you're looking at the data, you can say, okay, like the top two both had the yellow barcode. So this virus and this bacteria were in the same capsule and they were probably, in association or infecting, if there's an infection going on. So you can start to kind of link associations in different communities. And so that's kind of one application, but there's a lot of other different types of applications we're working on as well that I'm happy to talk about more, but I wanted to keep it short. But that's all. Thank you.
Thanks, Maureen, for talking to us about microfluidics and SPCs and what they can do. I'm putting into the chat the Joint Genome Institute. And so if you want to learn more about JGI, you are going to learn about it from our speakers today. But also, if you want to look online, you can do so. The next speaker we're going to bring is Yi Zhai on to our screen. So thank you so much for being here today. Yi also works within the Joint Genome Institute and is also going to talk about the biotech tools that they use here at the lab. So Yi, if you want to go ahead and share your screen. There we go. Thank you. I'll turn it over to you.
Hi everyone, my name is Yi. Yi as easy. So I'm a project scientist at Joint Genome Institute, part of the Lawrence Berkeley National Lab. It's a real pleasure to be here with you today and introduce a little bit about my research, which I like to call underground walkie-talkie. And actually they're just raggy podium distillation. This is the grass that I work on, and root transporters are the main character. And you can't just think like what I'm talking about is like how plants talk to microbes using chemistry. So I earned my PhD in cell molecular biology and development biology at UC Riverside. And I stayed there for like a few more years as postdoc. So back then I studied a group of organisms called oomycetes. So they are a whole kingdom that includes many plant and animal pathogens. So a lot of people think oomycetes are fungi, but they are not. So kind of like bats are not birds and dolphins are fish. We have this funny t-shirts about this. So the images that you see around me are the results from my past experiments. So during pandemic, I moved to LBNL and made a big shift from studying the plant pathogens to like beneficial microbes. So from bad guys to the good guys. So you will notice that a lot of my figures changed, but only one tool stuck here. So CRISPR. So that's it. I have introduced the title and showed you my CRISPR figures. So I guess that's the end of my talk today. Just kidding. So I will continue. So why do we care about beneficial microbes? As you all know, that climate change is already having serious effects on our environment. So it brings higher temperatures, less water, like bad soil quality, and like more extreme weather. So all this makes it like harder for plants to grow, especially food crops. So that means food security becomes like a big concern. So not just in developing countries, but everywhere. And there are like a lot of vulnerable communities already like feeling the impact, and many more people could be affected in the future. So how can we help plants survive and thrive in these challenging conditions? That's where beneficial microbes come in, and that's what we are trying to understand. So it has been known for a century that soil-borne microbes play a significant role in plant growth and health. So let's see what it looks like in the underground. The rhizosphere showed in this figure is a microbial like hotspot. So the microbe stimulates a lot of processes such as like nutrient cycling, denitrification, biocontrol of pathogens, later composition, and soil remediation. So that provides really important benefits to the plants, such as like enhance nutrient uptake, improve abiotic stress tolerance, promote plant health, growth, and last but not least, increase resistance against pests and pathogens. So the blowouts represent four main rhizosphere showing the microbiome processes. So since the soil microbes like play such critical role. So utilizing beneficial raw soil microbes will be a practical direction for sustainable agriculture. So in the meanwhile, research shows that the plants could also reshape their microbiome by secreting root exudates. Here comes the walkie-talkie transporters. So the exudates can be secreted through the transporter of the roots. So you can see the small little red dots, the red boxes, they are the transporters. So all the exudates can secrete it into the root rhizosphere to complete this network. So different kinds of exudates, they were transported by different families of transporters. So this is an illustration of the plant root with all kinds of root transporters that can transport different things. So what you're most familiar here, you can see sugars, proteins, and what else? Phenolics, and DNAs. So here are the overview of the strategies and approaches to help us understand like how transporter of the plants work to affect root exudates and also the microbiome. So we use like genetics approach, also metabolomics and microbiome analysis to study potential transporters that can change root exudates and the microbiome composition. So finally, we use this biochemical approach to study underlying mechanisms. So you can see like we have like single cell is pretty hot recently, and then metabolomic analysis, and then we can use 16S sequencing to study microbiome, and then again CRISPR tools. And these are the the fluorescence results picture from the microscope. And this is from microscope, but this is not plants. This is from protoplast. So to further confirm the functions of these transporters, so we have created knockout transporter mutants using CRISPR-Cas9 with a highly efficient transformation protocol developed by local lab at GGI. So you can see we dissect the embryo from the plants, and you can see this is the little single cell in single embryo. These are super tiny. They are like tinier than than my hair, than one piece of hair, and then they will be induced to callus, and then this callus will become bigger and bigger. So this become little plantlets, you can see here, and then they will help roots in the media, and then they become a whole big plants. So these are back in pandemic, like 3 years ago, I think, or 4 years ago. Like I work with 2 high school students. We are working on the CRISPR knockout transgenic plants. So are the tissue culture that you can see here. And these are 2 undergrad students from Alabama University. So they are like weighing. So they're weighing like they're weighing like gel powder, and then making DNA gel. So we're loading DNA gel here. And then we have students and and graduate students and we are working in the field at UC Davis. So here, like we have this like super cool wallpaper in our building. So I wish you have the chance to come to visit our building. Thank you for all your listening.
Thank you, Yi, for sharing with us, and great joke at the beginning. I think a couple of people enjoyed that. Our last speaker of today is Michael Kosicki, who has had some time at the laboratory, but also is now at ARC Institute. So Michael, I'll go ahead and allow you to share your screen for slides and share your journey with us.
All right. All right, yeah, thank you for having me. Great talks, all previous speakers. I will very briefly talk to you about what I've been doing the past few years. So I'm from Poland, Warsaw, and I did my undergrad in Denmark. Let's see if that goes anywhere. Yes, at the University of Copenhagen, looking at immunity, immunology. I won't talk about it that much. And I did my PhD at the University of Cambridge and Wellcome Sanger Institute in the UK, mostly focusing on CRISPR itself, and I will give you a little bit of insight into that. And about 6 years ago, I moved to LBL to do a postdoc. And just about a month ago, I moved where I'm right now, which is the ARC Institute. It's in the South Bay, just south of Stanford. So when I started my PhD, the kind of prevailing view of how Cas9 works, or CRISPR, is, it's a molecular scissors. It cuts the DNA, and then the cell tries to repair this break. Otherwise, it would kind of fall apart or become cancerous. And, most of the time it does a pretty good job and it repairs perfectly, and then Cas9 can come in and cut it again. And eventually it would make a mistake. And those mistakes, can be of all kinds of sizes. One cell can have, a loss of maybe 20 base pairs. Another one can have like a little insertion. But, the prevailing view was that all these mutations will be relatively small. They will be like up to maybe 20 base pairs. And we can use them, right? We can use them to break things basically in the genome. So we can break, each gene in the genome now in like a really high parallel way and find out what they do this way. But, we kind of assumed that this is going to be extremely localized, this damage. And in my PhD, I showed that this kind of image here aggregated over many, many cells, of like you just introduce those small mutations is actually kind of false, because 20% of all mutagenic outcomes of kind of regular Cas9 cutting the DNA and getting repaired is really large deletions of around thousands of base pairs. So like really orders of magnitude bigger than what people assumed. And, that had a lot of different consequences, one of which was that some companies that were doing gene therapies or proposing gene therapies at the time lost a little bit of their value. But more importantly, it really kind of pushed people to think about the safety and the outcomes that Cas9 does, and kind of pushed the field a little bit towards safer alternatives such as prime editing and base editing. And, in general, it kind of underscores that you get what you look for when you do science. If you look like at a very limited amount of space in the DNA, you would see small things, but you might miss kind of larger events. So, always like question your assumptions when doing science. I'll switch gears here and talk a little bit about what I did at Lawrence Berkeley Lab, and that's working and trying to understand how genomic elements called enhancers work. So if you take one thing from this particular talk is, what are enhancers? They're pieces of DNA that regulate or tell genes when and where to be active. So here's a brain enhancer. It tells the gene in here to be active in the brain. And there's another enhancer here. It's a limb enhancer. It tells the gene to be active in a limb. So this particular gene will be active in both. And, how is that important? Well, why is it important to study those things? Is obviously because mutations in those elements can, dysregulate genes and thus cause problems, anything from, limb malformation to brain issues. But the code of those elements is not very well understood. If there is a mutation in one of those, I can't really easily tell you what it will do. Whereas if there is a mutation in a gene, I can usually do it pretty well. Even you can do it with like a codon table. You can tell, oh, this amino acid changed. That will probably be bad, or this protein will not be produced anymore. That's probably bad. But with enhancers, we don't know. So how do we study them? In the lab that I've been at, Len Panacchio and Axel Weisels, well, first of all, we'll take the enhancer and add something to it that will report on its activity, some gene that produces color, basically. We'll inject that into a fertilized mouse egg, a single cell embryo, transfer that into pseudopregnant mice, wait a little bit for them to develop, and we get those beautiful embryos. They look like this. This is what we also looked like at some point about a few weeks after conception. And we also get, in this particular system, some color that tells us where the enhancer was active, and it can be multiple different tissues, as you can see. So now we're ready to, diagnose some cute patients like this Hemingway cat, which has, as you can see, 6 digits instead of 5. We know a little bit about limb development, and we know there's enhancers that are important for it. So we can test the enhancer activity of the kind of regular enhancer and an enhancer with a mutation that this particular cat has. And we'll see in this particular example that, the enhancer is active in the limb, but when you introduce the mutation, it gains activity in some other part of the limb. And, then you have a really good reason to now think that there is some relationship between the mutation and the actual phenotype, and you can maybe like make a mouse model or something. But, with this test, you've learned a little bit about it. Now, that's a little bit dull. You can do those tests for every single mutation, and there's like 3 billion possible mutations in the genome of just single base pair. You're never going to finish all your experiments. So you need to understand the code, as I mentioned, a little bit better so that you can predict it. And that's kind of what I set out to do for my postdoc. I thought, okay, let's take a bunch of enhancers with different activities across the body, and let's mutate them systematically. And I chose to mutate them in blocks of 12, doesn't really matter. And, then I would look at what the outcomes are. And, sometimes you mutate an enhancer, it gains activity. Sometimes nothing happens. Sometimes you lose activity. Okay, cool. If you do this across those 7 enhancers, you get a map, right? Like the map that I showed you at the beginning. It tells you something about each enhancers and where their kind of vulnerable points are and which points don't matter. And you've learned that like some of them are, have those elements which when mutated gain function and some of them don't. Okay, well, it's pretty variable. That's cool. But there's 2 problems with that. One, it's only 7 enhancers and we have like an estimated million of them. So we haven't really even scratched the surface. And second, we have this weird system, right? When we mutated things in blocks of 12, the map is a little bit blurry. And, what we really want is understand every single base pair and not some blocks of 12 base pairs. So for that, I reached out to another group that is actually at Stanford to use, everybody's favorite word, machine learning and artificial intelligence. So there is now models that can try and predict which part of the enhancer is important, and they can do it at single base pair level. They can tell you this letter, that's important, this letter, that's not important. Okay, well, that's cool. That's a good model. You can apply it to all million enhancers if you want. But does it actually work? That's model. Models are made sometimes in a vacuum, and they don't really correspond to anything real. So for my project, I actually could now take the actual results of my actual experiments in vivo and see how well they match the model. And you can see, most of the time, the models tell us the truth. They predict that, when they predict that something is important, it is important. On the other hand, when they predict that something isn't important, they aren't always right. Like in this case, right? It doesn't say anything in here, but there is definitely some function there. So we kind of really assess that systematically, and now we, know where the gaps are, know where the models can develop going into the future. But the cool thing about it is ultimately, they do help. So, for example, in here, you have some particular base pair that, could be mutated, like C to T, and you can ask the model what happens, and you can see that, this kind of activity goes away, and this is negative activity. So when you actually do the experiment, you see this massive gain of activity. So that is, I thought, really, really fun and interesting, and I'm really excited about, the future of machine learning in my field. And with that, I'll take the questions.
Great. Thank you, Michael. You can keep your camera on, and I'll invite our other 3 panelists to turn theirs on for our discussion now. I know there are quite a few questions coming in. If you want to also read more about Michael's work, I'm going to put the link to a recent Nature paper into the chat that you can read more that was recently published. Next, we're going to open our discussion. And the first question I want to share share with everyone is, you're doing all of this really innovative work that is new, right? That nobody's ever done before. That's the nature of research. But how do you deal with setbacks or failure, or readjust your goals for your project when, say, something doesn't work the first time? And so I'll start first with Michael and then go to Maureen, E, and then Shreya.
Sure. There's basically 2 answers to that. One of them is it's good to have something planned before your experiment fails. Like, whatever else you're gonna try, or if you have another project, try this, have this other project going. That really, I think, shields you to some degree from being just like, oh no, it failed and that's the end of the line and there's nothing else. And the second very practical advice is obviously, try to have a life. It helps to have something else going, like not have your entire self-worth dependent on science. That's it.
Yeah, I'll go next. Yeah, so as kind of in addition to the standard advice of that's very good that Michael said, I feel like something that's important that if failures sometimes give you good information, and so don't automatically think you're like it's the end of the world. Like I've had plenty of things where either I personally messed something up or something didn't happen and it ended up being a really useful piece of information. So sometimes like failures are there's a lot to learn from it. And that's actually helps propel the project into an interesting direction. So those types of setbacks can be end up being a positive thing. Not always, but sometimes. So like
So I think like science is all about like repetition. Just keep repeating. If everyone succeed on the first try, think about it. Like we would all have Nobel Prize, right? So when things don't work, I step back and I figure out what went wrong and adjust my strategy. So some experiments took me over 10 times to get it right. Um, but also I also believe like in that when sometimes we should let it go. Just sometimes you just need to go home, go shopping, or just go to sleep and like come back fresh, right? So next day it's gonna be a new day and science will still be there. If eventually like science is not your type, you can just switch like from science to other things like you like, right? So it's like it's not whole life. Yeah.
Yeah, and I definitely want to echo what Maureen was saying about how failure doesn't mean that nothing was learned. It's a chance to reevaluate and optimize whether it's your protocol or just like something else that you're working on. And so there's a lot to be gained from that. And then second, I think most importantly, it's remember to rely on the community around you. Science can feel really isolating at times when maybe you're the sole person working on a very niche topic. But remember that you have your lab mates, your advisor, your classmates, your friend, whoever to lean on. Everyone brings their own perspective to the table. And sometimes just like leaning on them, discussing with them can help come up with or help you come up with a very creative solution. And it's very okay to rely on your community. Science is very much about collaboration, even in times of failure.
Awesome. Thank you all for those first notes. And I think, Maureen, you did say it's great to learn what you like and you don't like. And that's what we try to say about experimentation. It can take you from research to just experiences that you have in life. It's all about getting to what you are really passionate about. So this can happen in the research laboratory too. For you all that are doing, you're working in different biotech tools that maybe the public doesn't understand all the time. What's a common misconception about the work that you do, especially as it concerns, say, maybe gene editing or working with genomes or working with fungi that people might not understand? So Shreya, I'm going to start back with you and then Yi and then Maureen and Michael. What are some misconceptions that people might have about your work?
Absolutely. As with many very little understood topics, people are scared of fungi. We generally hear about fungi in the context of diseases or spoiled food. Less known is that everyone is exposed to spores from very infectious fungi, fungal strains every single day, just because they have a ubiquitous presence in the air and in our food. Or maybe that fungi produce about 20% of global drug supply, including like antibiotics and cholesterol-lowering statins. And so many people have actually asked me as I've joined this field about whether I truly think it's safe to work with fungi. What if we end up building like really dangerous bioweapons? What if the fungi I work with contaminate my lungs? But again, the reality is that fungi is everywhere. And so they're in our medicine, in our food, in our ecology. And so that just makes demystifying them through research all the more important, and communication about the work that we do with the public all the more important, so that people can understand why we're researching what we do and develop like a healthy and knowledgeable relationship with that research.
So a common misconception is that using CRISPR means like we are creating GMOs or doing something dangerous or like unnatural. So in reality, we are often like just making like tiny targeted changes, like just switching like one base pair of the DNA. So just turning off like one single gene to understand what that gene does. So it's not changing other things probably. So it's more like editing a like a typo like in in a giant instruction manual than rewriting the whole the whole book. So it's a powerful tool for discovery, not just for engineering. And also in the lab, we have really strict regulations as well. We have to put transgenic plants or any microbes related things to autoclave beans, and then they will be killed by like really high temperature, so they won't go into the environment.
Yeah, so I guess kind of what you said was talking about, but add on to that slightly. I think there's this conception that I mean that we can engineer anything, like we have these fancy machines for it. Uh, I mean, I'm a fan of Jurassic Park, but like, we always, the kind of engineering they do there is very unrealistic, where they're putting, fish DNA in a on a frog, or whatever. And it's fun, but like, sometimes I can't get one bacterial species, a gene from one bacterial species to express in another, even though they're closely related. Like there's, a lot of regulatory elements are like really tight, and if you mess something up, everything gets screwed up. Like it's very complicated. And so what you were saying with like a lot of the changes we make are actually pretty small, and kind of creating any sort of like custom organism with like all these different features is extremely difficult, and I don't think anyone would want to put in the work to do the one thing. Usually it's pretty small, simple changes, nothing very science fiction like.
Just to add very briefly to the dinosaur question, the main problem is we just don't have dinosaur DNA that is like intact. It's like so old that it's just all fallen apart. We could create something new that like resembles a dinosaur maybe someday, but like definitely not actual DNA. We just don't have it. But yeah, I think the main misconception people have in terms of editing is that human embryo editing could even be like necessary. Like when you think about it somewhat more deeply, there's basically almost no medical need whatsoever ever to edit like a complete human embryo. Like 90%, 95% of cases, you can deal with like genetic diseases if you, of course, like are so inclined by using in vitro fertilization and selecting the embryos. I know that's like a problematic thing in other contexts, but when people think about it, they basically think of those cases. I'm like, no, you don't need to like do CRISPR in this particular case. You can just go around it completely.
Awesome. Thank you all for sharing that. One question from the chat is, what is one moment of your research where you found success and had this feeling of what I am doing is worth it? So I'll allow you to think for a second, but Maureen, I'm going to put you on the spot first, and then Yi, and then Michael, and then Shreya.
I guess kind of like on a personal point. I mean, I don't think it's like going to change the world, but I feel like in grad school, there's kind of this point, at least for me, where you're kind of diverging from your kind of supervisor's ideas, and you're trying to create your own. And kind of that first project that you're doing is, these are all of your ideas. It's everything's all in your own. And so kind of trying that first experiment and like actually having it work. And it's kind of this, I don't know, confidence building experience where it's like, oh, like what I thought, like I did all this background research with all these ideas on my own. I designed everything myself. This kind of my first, everything's on my own and it worked. So I feel like for me, that was kind of a big moment of, I guess, becoming kind of like an independent science, even though we work with scientists in all our projects. But like that, I guess that was the first thing that popped in my head of just like I designed the screen because I was looking for certain things and like it actually worked and like gave me what I expected. So that was kind of a nice simple moment, but kind of like an important moment in my graduate training.
I actually like back to the first question. Actually, when you have failure, like after you have failures, you adjust your strategy, right? And then the time that it's successful, so that feels the best because it's like a debugging, like when you do programming work, right? And then you just work on the program, work on the codes, like after 3 days and 3 nights, okay, oh, this worked out and this program just run. So that feels fantastic, fantastic. It means like what I have like done in the past days, it just worked, and then I proved myself, and then have to like, and also I solved the problem for other people, so that feels the best.
Yeah, I was extremely lucky and privileged to like have, a very like big breakthrough during my PhD. And yeah, there was a very like clear moment where I was doing, some control experiment. It wasn't even like to test the idea that Cas9 causes like large deletions and other things. I was just doing a control, and like the control was so off. And I was just like, oh, this definitely looks interesting. And if it's true, which took, another like year and a half of work, like that would be a pretty big finding. And I was just hoping nobody's gonna scoop me. But yeah, that's obviously, nice. But I think those moments like scientists get like, once in a lifetime if they're lucky. And I'm, just been very lucky again. Yeah.
Yeah, for me, I think one of the biggest successes that I've learned from is not necessarily a success in the science, but a success in understanding my need space. So I used to work in more of the diagnostic side, so I know very different. And so we were creating some diagnostics specifically for colorectal cancer, and we had an idea we were testing out. We had a lot of issues with the chemistry of creating the molecule that we were hoping to create. And so part of our process of creating this diagnostic was also to talk to the people we're trying to serve. So talk to these colorectal cancer patients, talk to some other folks with inflammatory bowel disease who we were hoping to also serve. And so it was moments of conversation with these people where we understood just how the current market wasn't serving them and just how badly they needed change in the market that were the most motivating moments for us, the moments where they were really excited about what we were doing. And so I'd say that, the more you understand about your problem, the more you talk to stakeholders within the problem you're trying to solve, the more motivation you can find, and the more like amazing that success will end up feeling too.
Okay, so we're coming up on the hour. I am trying to repost, and I know our speakers are kind of chatting in getting to some questions in the chat as well. Our final question is kind of a little bit of what someone asked in here of just what changes would you have made in your experiences in science if you had to go back and give yourself advice? So what are some of the things that you might have told yourself back when you were in high school or starting off your early college career to make your experience, say, maybe just the path a little bit easier? So Yi, I'm going to start off with you, and then Shreya, and then Michael, and then Maureen to finish.
I will say like try to be a more organized person. So have like more organized notes and design like experiments like earlier before you start anything. So spend more time like on reading than actually like repeating the work. And then if I back to high school, okay. So I suggest like after if I have the chance, I would like to have like a gap year after like high school or after college, like to do more rotations in different area of the labs. So not just staying in one specific area, field, right? So try different things like plants or animal cells or like cancer cells, or like cell lines, or like dissection, or even some people like dissecting animals. So just try it in different labs so you will find out what you like or what you don't like. So give yourself like more opportunities.
And for me, I would say definitely invest effort and time into your communication skills early on. Science today, especially the biotechnology field, with many questions about the future of prominent like federal science organizations, and with many people fearing like modern medicines like vaccines. It's like a very like tumultuous and divisive field. And it wasn't too long ago that I was in your shoes trying to figure out what it means to like make a difference as a scientist and how to emit the most good into the world. And throughout these past few years, I've definitely learned that your science is most effective when the people that it affects can make informed decisions about how to engage with it. Misinformation is going to lead to fear, which is then going to close down doors and prevent new technologies from being adopted. So no matter how good your science is, if people are scared of it or are not willing to engage with it, it won't really make it out of the lab. And so knowing how to communicate your science to the public and sort of dispel those fears is a very important skill to have.
Yeah, as usual with these advice, there's just one advice at the end of the day, which is like listen to yourself, try to challenge yourself, but also to have fun. But specifically for myself, I feel like I kind of early on, was more prestige oriented, that like it's even necessary at all. Like when you go into an undergrad, you can go to like a big university and try to join a big lab, and you're gonna be like a small fish in a pond, and that has its advantages if you want to challenge yourself. That's one way of doing this. But like I don't think it's really that important early on. Like early on, it's important to like try things out, see what you like. Again, as reflecting on other people's responses. So don't be like fully driven by that super early on.
Yeah, I can finish. Yeah, I mean, I'm kind of at the perspective of that, like I like where I am now, and if I change anything, I may end up somewhere different. All the things I would think of like, oh, I should have done that. I started my PhD at 22, like I was young, and so a lot of the things that I, would have changed, it was just a function that I was young. And I just didn't have as much experience, in terms of just, running projects and things like that. And so I don't know if I would necessarily, I don't think a 22-year-old would listen to me. I might have been too stubborn, but I think because I've interacted with a lot of students in the Bay Area, and I think an overall like whelming thing is that they are very motivated and it's very impressive, but sometimes there's a little too like, I don't know, pressure to kind of figure it out at the beginning. And I was the opposite of that, which has its pros and cons. Like I kind of just tried a bunch of different things and just kind of floated around, and it happened to work out. But there's definitely a scale of like, I kind of just I asked someone an opportunity. I was like, yeah, I'll try it. Let's see what happens. And I could think if I was going to kind of change anything is maybe have a slightly more focused goals, or at least a bit more conscious of it instead of just kind of seeing where the wind takes me. So I think if there's kind of a middle ground, I think younger me could have learned something from a lot of the very ambitious Bay Area students, but they could have also maybe been a little bit, you don't have to figure everything out. Like, you can try a bunch of different things, and just switch around. You hopefully have a long life, there's time to try things. So, that's kind of the roundabout answer.
All right. Thanks, Maureen. And thanks everybody for, again, I think just reiterating, try again. That's in research and in life. Be open to opportunities. Make sure your science is able to be transferable and communicative, or you're able to communicate your science to the people that it really affects. All of those are great things to take away from today. And I want to thank our panelists. Hopefully the chat can thank them as well. And for those who are on site in our experiences in research program and the other programs here, hopefully they'll see you walking through the halls and be able to say hello at some point. Thank you to everybody else who's online. We will see you next week, or not next week because it's a holiday, so the following week with a panel on microelectronics. So thank you all so much, and thank you again to the panel.